Fetal development during pregnancy is measured in a number of weeks after fertilization. An average human pregnancy lasts for about 280 days or 40 weeks from the date of the last menstrual period, or LMP. Traditionally, it has been calculated as 10 lunar months, or in terms of the modern calendar, 9 months. Fertilization of the egg by the sperm, however, usually occurs 14 days after the last period. Thus, the average actual duration of a human pregnancy or gestational period is 280 days minus 14 days, which equals 266 days. The 3 stages of fetal development during pregnancy are as follows: the pre-embryonic stage, the embryonic stage, and the fetal stage. The pre-embryonic stage is fertilization through the 2nd week of pregnancy. The embryonic stage is the end of the 2nd week through the 8th week of pregnancy. The fetal stage is the end of the 8th week until birth. Fetal circulation is a significant aspect of fetal development that spans all 3 of these stages. The pre-embryonic stage begins with fertilization, also known as conception. Fertilization is the union of the ovum and the sperm, which is the starting point of pregnancy. Fertilization typically occurs around 2 weeks after the last normal menstrual period in a 28-day cycle. Fertilization requires a timely interaction between the release of the mature ovum at ovulation and the ejaculation of enough healthy, mobile sperm to survive the hostile vaginal environment through which they must travel to meet the ovum. All things considered, the act of conception is difficult at best. To say merely that it occurs when a sperm unites with the ovum is overly simplified, because this union actually requires an intricate interplay between the hormonal preparation and overcoming an overwhelming number of natural barriers. A human being is truly an amazing outcome of this elaborate process. Prior to fertilization, the ovum and the spermatozoon undergo the process of meiosis. The primary oocyte completes its first meiotic division before ovulation. The secondary oocyte begins its second division just before ovulation. Gametogenesis is the process by which gametes, which are the ovum or sperm cells, are produced to initiate the development of a new individual. The gametes must have a haploid number of chromosomes, 23, so that they come together to form the normal human diploid number of chromosomes, which is 46. The formation of gametes by the process of meiosis is known as gametogenesis. Spermatogenesis is the process of one spermatogonium giving rise to 4 spermatozoa. In oogenesis, from each oogonium, 1 mature ovum and 3 abortive cells are produced. The chromosomes are reduced to 1/2 the number of the characteristic for the general body of cells of the species. In humans, the number in the body cells is 46, while the chromosome number in a mature spermatozoon and secondary oocyte is only 23. Although each millimeter of ejaculated semen contains more than 200 million sperm, only 1 is able to enter the ovum to fertilize it. All of the other spermatozoa are blocked by the clear protein layer called the zona pellucida. The zona pellucida disappears in about 5 days. Once the sperm reaches the plasma membrane, the ovum resumes meiosis and forms a nucleus with half the number of chromosomes. When the nucleus from the ovum and the nucleus from the sperm make contact, they lose their respective nuclear membranes and combine the maternal and paternal chromosomes together. Because each nucleus contains a haploid number of chromosomes, 23, this union restores the diploid number to 46. The resulting zygote begins the process of a new life. The genetic information from both the ovum and the sperm establishes the unique physical characteristics of the individual. Sex determination is also determined at fertilization and depends on whether the ovum is fertilized by a Y-bearing sperm or an X-bearing sperm. An XX zygote will become a female, and an XY zygote becomes a male. In the inheritance of gender, each ovum contains 22 autosomes and an X chromosome. Each spermatozoon or sperm contains 22 autosomes and either a Y chromosome or an X chromosome. The gender of the zygote is determined at the time of fertilization by the combination of sex chromosomes of the sperm, whether it bears an X or a Y chromosome, and the ovum, which always carries an X for the sex chromosome. Fertilization takes place in the outer third of the ampulla of the fallopian tube. When the ovum is fertilized by the sperm, now called a zygote, a great deal of activity immediately takes place. Mitosis or cleavage occurs as the zygote slowly is transported into the uterine cavity by the tubal muscular movements of the fallopian tube. After a series of 4 cleavages, the 16 cells appear as a solid ball of cells called a morula. The word morula means little mulberry. The morula reaches the uterine cavity about 72 hours after fertilization. Multiple fetuses can occur at the time of fertilization when more than one ovum is fertilized. Identical twins, also called monozygotic twins, occur when one fertilized egg splits and develops into 2 or occasionally even more fetuses. The fetuses usually share one placenta. Identical twins have the same genes, so they generally look alike and are the same sex. Fraternal twins, also known as dizygotic twins, develop when 2 separate eggs are fertilized by 2 different spermatozoa. Each twin usually has its own placenta. Fraternal twins are just like any other siblings, sharing about 50% of their genes, and they can be different sexes. They generally do not look any more alike than a usual brother and sister would. Fraternal twins are more common than identical twins. With additional cell divisions, the morula divides into specialized cells that will later form fetal structures. Within the morula, an off-centered, fluid-filled space begins to appear, transforming it into a hollow ball of cells called a blastocyst. The inner surface of the blastocyst will form the embryo and the amnion. The outer layer of cells surrounding the blastocyst cavity is called the trophoblast. Eventually, the trophoblast develops into one of the embryonic membranes, called the chorion, which helps to form the placenta. At this time, the developing blastocyst needs more food and oxygen in order to keep growing. The trophoblast attaches itself to the surface of the endometrium for further nourishment. Normally, implantation occurs in the upper uterus, called the fundus, where a rich blood supply is available. This area also contains strong muscular fibers, which clamp down on blood vessels after the placenta separates from the inner wall of the uterus. Additionally, the lining is thickest here, so the placenta cannot attach so strongly that it remains attached after birth. Concurrent with the development of the trophoblast and implantation, further differentiation of the inner cell mass occurs. Some of these cells become the embryo itself, and others give rise to membranes that surround and protect the embryo. The 3 embryonic layers of cells begin to form. These are the ectoderm, the mesoderm, and the endoderm. The ectoderm forms the central nervous system, special senses, skin, and glands. The mesoderm forms the skeleton, the urinary, circulatory, and reproductive organs. The endoderm forms the respiratory system, along with the liver, the pancreas, and the digestive system. These 3 layers are formed at the same time as the embryonic membranes, and all tissues, organs, and organ systems develop. Despite the intense and dramatic activities going on internally to create a human life, many women are unaware that pregnancy has begun. Several weeks will pass before even one of the presumptive signs of pregnancy, such as missing the first menstrual period, will take place. The embryonic stage of development begins at day 15 after conception and continues through week 8. Basic structures of all major body organs and the main external features are completed during this time period, including internal organs. The embryonic membranes begin to form around the time of implantation. The chorion consists of trophoblast cells and a mesodermal lining. The chorion has finger-like projections called the chorionic villi on its surface. The amnion originates from the ectoderm germ layer during the early stages of embryonic development. It is a thin, protective membrane that contains amniotic fluid. Alongside of the amnion, a yolk sac develops as a secondary cavity around day 8 or 9 after conception. The yolk sac aids in transferring maternal nutrients and oxygen to the embryo during the 2nd and 3rd weeks of gestation, when the development of the uteral placenta circulation is still underway. As the pregnancy progresses, the yolk sac atrophies and is incorporated into the umbilical cord. As the embryo grows, the amnion expands until it touches the chorion. The two fetal membranes form the fluid-filled amniotic sac that protects the floating embryo. Amniotic fluid surrounds the embryo and increases in volume as the pregnancy progresses, reaching approximately a liter at term. Amniotic fluid is derived from two sources: fluid transported from the maternal blood across the amnion and fetal urine. Its volume changes constantly as the fetus swallows and voids or urinates. Sufficient amounts of amniotic fluid help to maintain a constant body temperature for the fetus, permits symmetric growth and development, cushions the fetus from trauma, allows the umbilical cord to be relatively free from compression, and promotes fetal movement to enhance musculoskeletal development. Amniotic fluid is composed of 98% water and 2% organic matter. It is slightly alkaline and contains albumin, urea, uric acid, creatine, bilirubin, lecithin, sphingomyelin, epithelial cells, vernix, and fine hair called lanugo. Amniotic fluid is essential for fetal growth and development, especially fetal lung development. It is dynamic, constantly changing as the fluid moves back and forth across the placental membrane. The volume of amniotic fluid is important in determining fetal well-being. It gradually fluctuates throughout the pregnancy. The rate of change of the amniotic fluid depends on the gestational age. During the fetal stage, the increase is 10 milliliters per week, and it increases to between 50 and 60 milliliters per week at 19 to 20 weeks gestation. The volume is at maximum at 30 weeks gestation before it undergoes a gradual decrease by term. Alterations in the amniotic fluid can be associated with problems in the fetus. Too little amniotic fluid, less than 500 milliliters at term, is termed oligohydramnios and is associated with uteroplacental insufficiency and fetal renal abnormalities. Too much amniotic fluid, over 2,000 milliliters at term, is termed hydramnios. Hydramnios is associated with maternal diabetes, neural tube defects, chromosomal deviations, and malformations of the central nervous system or gastrointestinal tract that prevent normal swallowing of amniotic fluid by the fetus. While the placenta is developing during the end of the 2nd week, the umbilical cord is also formed from the amnion. The umbilical cord is the lifeline from the mother to the growing embryo. It contains one large vein and two small arteries. Wharton's jelly, which is a specialized connective tissue, surrounds these 3 blood vessels in the umbilical cord to prevent compression, which would cut off fetal blood and nutrient supply. At term, the average umbilical cord is about 22 inches long and about 1 inch wide. The precursor cells of the placenta are the trophoblasts. These first appear 4 days after fertilization as the outer layer of cells of the blastocyst. These early blastocyst trophoblasts differentiate into all of the cells that form the placenta. When fully developed, the placenta serves as the interface between the mother and the developing fetus. Most commonly, the placenta develops at the uterine fundus. As early as 3 days after conception, the trophoblasts make human chorionic gonadotropin, or HCG, which is the hormone that ensures that the endometrium will be receptive to the implanting embryo. During the next few weeks, the placenta begins to make hormones that control the basic physiology of the mother in such a way that the fetus is supplied with the nutrients and oxygen needed for growth. The placenta also protects the fetus from immune attack by the mother, removes waste products from the fetus, induces the mother to bring more food to the placenta, and, near the time of delivery, produces hormones that ready fetal organs for life outside the uterus. Theoretically, at no time during pregnancy does the mother's blood mix with fetal blood cells, because there is no direct contact between their bloods. Layers of fetal tissue always separate the maternal blood and fetal blood. These fetal tissues are called the placental barrier. Materials can be interchanged only through diffusion. The maternal uterine arteries deliver the nutrients to the placenta, which in turn provides nutrients to the developing fetus. The mother's uterine veins carry fetal waste products away. The structure of the placenta is usually completed by week 12. The placenta is not only a transfer organ, but a factory as well. It produces several hormones necessary for normal pregnancy. These include human chorionic gonadotropin, or HCG, human placental lactogen, HPL, human chorionic somatomammotropin, or HCS, estrogen, and progesterone. HCG, or human chorionic gonadotropin, preserves the corpus luteum and its progesterone production so that the endometrial lining of the uterus is maintained. Human chorionic gonadotropin is also the basis for pregnancy tests. Human placental prolactin, or human chorionosomatomammotropin, modulates fetal and maternal metabolism, participates in the development of maternal breasts for lactation, and decreases maternal glucose utilization, which increases glucose availability for the fetus. Estrogen causes enlargement of a woman's breasts, uterus, and external genitalia, and stimulates myometrial contractility. The progesterone maintains the endometrium, decreases the contractility of the uterus, stimulates maternal metabolism and breast development, and provides nourishment for early conception. The umbilical cord also produces relaxin. Relaxin acts synergistically with progesterone to maintain the pregnancy. It also causes the relaxation of pelvic ligaments and softens the cervix in preparation for birth. The placenta acts as a pass-through between the mother and the fetus, not a barrier. Almost everything the mother ingests, including food, alcohol, and drugs, passes through to the fetus. This is why it is so important to advise pregnant women not to use drugs, alcohol, or tobacco. During the embryonic stage, the embryo grows rapidly, as all organs and structures are forming. During this critical period of differentiation, the growing embryo is most susceptible to damage from external sources, including substances that can cause birth defects such as alcohol and drugs, infections, including rubella and cytomegalovirus, radiation, or nutritional deficiencies. The fetal stage is the time from the end of the 8th week until birth. It is the longest period of prenatal development. During this stage, the embryo is mature enough to be called a fetus. Although all major systems are present in their basic form, dramatic growth and refinement of all organ systems takes place during this period. The circulation through the fetus during the uterine life differs from that of a child or an adult. In the extrauterine world, oxygenation occurs in the lungs, and oxygenated blood returns via the pulmonary veins to the left side of the heart to be ejected by the left ventricle into systemic circulation. In contrast, fetal circulation oxygenation occurs in the placenta, and the fetal lungs are non-functional as far as the transfer of oxygen and carbon dioxide is concerned. Oxygenated blood travels from the placenta to the fetus's systemic circulation through a series of shunts. Fetal circulation involves the circulation of blood from the placenta to and through the fetus and then back to the placenta. A properly functioning fetal circulatory system is essential to sustain the fetus. Before it develops, nutrients and oxygen diffuse through the extraembryonic column and the yolk sac from the placenta. As the embryo grows, its nutrient needs increase. Thus, the circulation must develop quickly and accurately. 3 shunts are present during fetal life: the ductus venosus, the ductus arteriosus, and the foramen ovale. The ductus venosus connects the umbilical vein to the inferior vena cava. The ductus arteriosus connects the main pulmonary artery to the aorta. The foramen ovale is the anatomical opening between the right and left atrium. The blood with the highest oxygen content is delivered to the fetal heart, neck, head, and upper limbs, while the blood with the lowest oxygen content is shunted towards the placenta. The oxygenated blood is carried from the placenta to the fetus via the umbilical vein. About half of this blood passes through the hepatic capillaries, and the rest flows through the ductus venosus into the inferior vena cava. Blood from the vena cava is mostly deflected from the foramen ovale into the left atrium, then to the left ventricle, into the ascending aorta, and then on to the head and upper body. This allows the fetal coronary circulation and brain to receive the blood with the highest level of oxygen. Deoxygenated blood from the superior vena cava flows into the right atrium, the right ventricle, and then the pulmonary artery. Because of high pulmonary vascular resistance, only a small percentage, about 5 to 10% of the blood in the pulmonary arteries flows to the lungs. The majority is shunted through the patent ductus arteriosus and then to the descending aorta. The fetal lungs are essentially non-functional because they are filled with fluid, making them resistant to incoming blood flow. They only receive enough blood for proper nourishment. Finally, two umbilical arteries carry the unoxygenated blood from the descending aorta back to the placenta. At birth, a dramatic change in the fetal circulatory patterns occurs. The foramen ovale, ductus arteriosus, ductus venosus, and umbilical vessels are no longer needed. With the newborn's first breath, the lungs inflate, which leads to an increase in blood flow to the lungs from the right ventricle. This increase raises the pressure in the left atrium, causing a one-way flap on the left side of the foramen ovale called the septum primum to press against the opening, creating a functional separation between the two atria. Blood flow to the lungs increases because blood entering the right atrium can no longer bypass the right ventricle. As a result, the right ventricle pumps blood into the pulmonary artery and onto the lungs. Typically, the foramen ovale is functionally closed within 1 to 2 hours after birth. It is physiologically closed by 1 month by deposits of fibrin that seal the shunt. Permanent closure occurs by the 6th month of life. The ductus venosus, which links the inferior vena cava with the umbilical vein, usually closes with the clamping of the umbilical cord and inhibition of blood flow through the umbilical vein. This fetal structure closes by the end of the first week. The ductus arteriosus constricts partly in response to the higher arterial oxygen levels that occur after the first few breaths. This closure prevents blood from the aorta from entering the pulmonary artery. The embryonic stage of development begins at day 15 after conception and continues through week 8. Basic structures of all major body organs and the main external features are completed during this time period, including internal organs. By week 3, the brain, spinal cord, and heart begin to develop, along with the gastrointestinal tract. Here, the neural tube forms, which later becomes the spinal cord. Also, leg and arm buds begin to appear and grow out from the small body. In week 4, the brain begins to differentiate and the limb buds grow and develop more. In week 5, the heart now begins to beat a regular rhythm. Beginning structures of the eyes and ears are noticeable. Some cranial nerves are now visible and muscles connect to nerves. In week 6, the formation of the lungs begins and fetal circulation is established. The liver begins producing red blood cells and the brain develops further. Here, the primitive skeleton forms and the central nervous system is now detectable with brain waves. In week 7, we see a straightening of the trunk. Nipples and hair follicles form, along with elbows and toes. Arms and legs begin to move more frequently, and the diaphragm is formed. The mouth and lips become visible along with early tooth buds. In week 8, there is a rotation of the intestines, and facial features continue to develop. The heart development is now complete, and the embryo begins to resemble a human being. In weeks 9 through 12, Sexual differentiation continues. Buds for all 20 temporary teeth are now laid down, and the digestive system shows activity. In this stage, the head makes up nearly half the size of the fetus. The face and neck are well formed, and the urogenital tract completes its development. Red blood cells are produced in the liver, and urine begins to be produced and excreted. The fetal gender can be determined by week 12 using an ultrasound, which will also reveal limbs that are long and thin and digits that are well formed. In weeks 13 through 16, a fine hair called lanugo develops on the head. The fetal skin is almost transparent as bones become harder. The fetus makes active movements, and sucking motions are made with the mouth. The fetus swallows amniotic fluid, and external genitalia are recognizable at this stage. Fingernails and toenails are also present as the fetus's weight quadruples. It is usually during this phase that fetal movement, also known as quickening, is detected by the mother. Weeks 17 through 20 show a rapid brain growth with fetal heart tones that can be heard with a stethoscope. The kidneys will continue to secrete urine into the amniotic fluid. The vernix caseosa, which is a white greasy film, now covers the fetus. The eyebrows and head hair may appear, and brown fat is deposited to help maintain the fetus's temperature. Muscles are well developed, and nails are present on both fingers and toes. During weeks 21 through 24, eyebrows and eyelashes are well formed. The fetus has a hand grasp and startle reflex. Alveoli begin forming in the lungs, and skin remains translucent and red. The eyelids still remain sealed, and the lungs begin to produce surfactant. During weeks 25 through 28, the fetus reaches a length of about 15 inches. There is a rapid development of the brain, and eyelids are able to open and close. The nervous system is now able to control some of the functions of the fetus. Fingerprints are also set. Subcutaneous fat is now visible under the skin. Blood formation shifts from the spleen to bone marrow, and the fetus usually assumes a head-down position. In weeks 29 to 32, there is a rapid increase in the amount of body fat. There is an increased central nervous system control over body functions, and rhythmic breathing is now established. However, the lungs are not fully mature. Pupillary light reflex is now present, and the fetus stores iron, calcium, and phosphorus. During weeks 33 through 38, the testes are in the scrotum of the male fetus. Lanugo begins to disappear. The fetus has a good, strong hand grasp reflex and increases in body fat. Earlobes are formed and firm. Fingernails reach the end of the fingertips, and small breast buds are present on both sexes. The mother supplies the fetus with antibodies against disease. The fetus is considered full term at 38 weeks, when the fetus fills all the available space in the uterus.